So this table is a good example of some of the many tests that we can run to check for abnormalities in the blood. One example here is macrocytic anemia, and that is a type of anemia that causes large red blood cells. Macro meaning large and cytic meaning cell. Like other types of anemia, macrocytic anemia also often means that the red blood cells are low in hemoglobin. Hemoglobin is the oxygen carrying molecule, and so that also means that we are low in oxygen getting to our tissues, supplying our tissues. This can be caused by deficiencies of things like B12 or folate. Microcytic anemia, micro meaning small, so is the presence of small, often hypochromic. Hypo means below, and chromic refers to color. So these will be small, pale red blood cells, and these will usually have a low MCV, so mean corpuscular volume. So for microcytic anemia, you would look at mean corpuscular volume. For macrocytic anemia, you would look at mean corpuscular hemoglobin, so just a slight difference there. Pay attention to the normal values that go along with the particular test that you are running, because some will have different ranges depending on the distributor or the person, the company that makes the test. So they can have slight differences here. Iron deficiency anemia is the most common cause of microcytic anemia. Total iron binding capacity values above 450 milligrams per deciliter, or micrograms per deciliter, usually mean there's a low level of iron in your blood, and that's usually due to things like a low iron in the diet, excessive bleeding due to things like menstruation or peptic ulcers, or even the increase in blood volume needs due to pregnancy. So this is why sometimes pregnant women need to take iron as well, part of the reason, and the folate. So both of these tests here relate to the size of your red blood cells, and you can read the rest of these yourself and kind of get a little familiar with these other tests. All right, this is a very important chart that you need to know. I would highly recommend actually drawing this out using some of these pictures here that you can see to help you to remember these. You need to know the percentages and everything. Blood composition is a complex mixture of formed elements and plasma. So under the formed elements here, let's break that down in percentages here. Formed elements account for about 45% and plasma about 55% normally. In the formed elements, we have platelets, red blood cells, and white blood cells. White blood cells, while only 0.1% of the components here, is a very important part of your immune system. Platelets here, which are 4.8%, have a function for clotting, so they're important as well. And then, of course, red blood cells compose about 95%, a little over 95%. Plasma contains all of the substances that are needed in the body. So the cardiovascular system is like a transport system. It carries things to and fro to different little cities in our body, and these little cells in our body that need different things, all right? So it contains and carries nutrients and vitamins, hormones, gases like oxygen that needs to go to cells, and carbon dioxide that needs to be released, other waste products, and even water and electrolytes. You can see the white blood cells are broken down into these 5 types here. This is important as well. You do need to know these percentages. This is usually an essay in class. The neutrophils are 54 to 62%, eosinophils 1 to 3, basophils less than 1, monocytes 3 to 9, lymphocytes between 25 and 33. You can see we're going to look at some of the different types of proteins as well. We are going to break those down and look at those. All right, so let's take a look at where blood cells originate from. Blood cells originate in the red marrow from hemocytoblast or hematopoietic stem cells. Stem cells are cells that can specialize or differentiate into other types of cells or more stem cells, so they're pretty handy. We have two types of stem cells that they can become are going to be lymphoid and myeloid. These are going to respond to hematopoietic growth factors that can turn specific genes on and off, which determines what the stem cell will eventually become. This also can make the different formed elements of the blood. Thrombopoietin, which is TPO, can cause what we call a megakaryocyte to proliferate, or to rabbits proliferate, right? So they can replicate abundantly and quickly, and those can actually become platelets later on down the line. They can break apart. So this particular slide here shows us all the different paths that a hematopoietic cell can take to differentiate into all the different cells that we have. So if we start with the hemocytoblast, what is the correct sequence of differentiation for a red blood cell? And so you're going to follow this path here that I have outlined with the red circle. So you would start with a hematopoietic stem cell, move on down to the myeloid stem cell, and that's going to move on down through to the erythroblast, reticulocyte, on down all the way to the erythrocyte. So what about a macrophage? Well, we could find a macrophage down here and follow this path here. Hematopoietic stem cell, monoblast, monocyte, which can become a macrophage. And so you can see how all of these differentiate into the different cells that we have in our blood. All right, so let's look at the different characteristics of red blood cells, which we call erythrocytes. I've already stated they are biconcave discs, so they're kind of indented in the middle. The shape is very important because remember, structure determines function. The structure increases the surface area for gas diffusion and allows maximum transport of gases, particularly of oxygen. So it is also the right size to fit through our tiny little capillaries, which is very important for distribution. A red blood cell is about 1/3 hemoglobin, which is what gives us that red color, and 2/3 water, and then it has electrolytes and enzymes as well. Oxyhemoglobin is hemoglobin that is bound with oxygen, that has that characteristic red color. Deoxyhemoglobin is going to be darker bluish in color inside the blood vessels. These have nuclei during early development, but once they mature, they no longer have nuclei. They cannot make their own proteins, and they no longer divide, which is kind of unusual for a cell. Red blood cells make ATP through glycolysis, but they do not have mitochondria. So we take a red blood cell count in order to count the number of red blood cells in a millimeter cubed or microliter of blood. This can vary depending on age and health. Typical ranges for males, you can see here listed at the top, 4 million to 6 million. Below that we have adult females, and then ranges for children as well. This reflects again the oxygen carrying capacity of the blood, so these are important. The number of red blood cells can actually increase for several days following strenuous exercise or an increase in altitude. Red blood cells are routinely looked at to help diagnose and evaluate various diseases. Changes in the number of these can affect health by affecting that oxygen carrying capacity. So this is important. So what are the factors that control red blood cell production? This process is called, the process of red blood cell formation is called erythropoiesis. This actually occurs in the yolk sac, the liver, and the spleen. After birth, they are almost exclusively only produced by red bone marrow. However, interesting enough, we have recently found that the human intestine may provide up to 10% of blood cells in circulation from its own reservoir of blood-forming stem cells. This was discovered when we had transplants of intestinal tissues that were producing blood cells. So this was a pretty astounding fact that we've learned in, say, the past 10 years. So that's what led us to that understanding. So low blood oxygen is going to, this process is going to cause, we're looking at this diagram here, low blood oxygen is going to cause the kidneys to release erythropoietin, which we call EPO. That is going to then stimulate red blood cell production. And you can look at this diagram in your textbook. This is a negative feedback mechanism. Remember, negative feedback serves to return things to normal, right? Positive feedback moves them further away. Negative feedback wants to bring something back to a normal range or value, all right? And so you can see the path there going from hemocytoblast that we looked at earlier to erythroblast as it's maturing onto reticulocyte and eventually an erythrocyte. So within a few days, many new blood cells appear in the circulating blood. All right, so dietary factors that can affect our red blood cell production. What is important in the diet? B vitamins, folic acid, and iron are all very vital to red cell production. B12 and folic acid are needed for cell growth and division early on. Since hematopoietic cells divide a lot, they can easily become deficient in these vitamins. Lack of B12 is often due to absorption issues because the cells in the stomach secrete what we call an intrinsic factor, which is required to absorb and use that vitamin B12. The form of anemia caused by a lack of dietary iron, remember, is iron deficiency anemia. Vitamin K is also needed. This is for the synthesis of prothrombin. We need vitamin C, which can increase absorption of iron in the digestive tract. Drinking orange juice with a meal is a good way to actually boost iron absorption. Drinking tea with a meal can actually reduce absorption of iron because it has tannic acid in the tea, which binds to dietary iron and prevents its absorption. So understanding this is important. That's something a tip you could give to patients that are taking iron or people that need B12. So looking a little more at anemia, which is abnormal red blood cells, we have a light micrograph here on the bottom left with erythrocytes and that have a central pallor or paleness to them. All right, here on the right you can see that that they're wider in the center than some of the other ones. In iron deficiency anemia, which is B, you can see you have actually sickled erythrocytes, and there's an arrow pointing to this, okay? And so you can see that these are really kind of white, and there's an arrow pointing to this one. I think I said right, I meant on the left. And so those are the ones that are pale, okay? They have pallor. And then this one is sickled, so sickle cell anemia. And you will have a video that you need to watch on sickle cell anemia. And it's very important that you do watch that because you will be tested over some of the information on that, all right? And we'll go into more details about sickle cell anemia as well. So anemia is basically a deficiency of red blood cells or reduced hemoglobin, which then reduces the carrying capacity of oxygen, which causes pallor not only of the red blood cells but also in our skin. That's why we get pale, and fatigue. And here is a nice little chart over here that talks about some different types of anemia. I would read about the cause and effect there in the name and just be generally familiar with these. All right, so as red blood cells get older, they become more fragile and can be damaged when they are passing through capillaries. They can rupture passing through the spleen. Heme can be released from the hemoglobin in an older red blood cell and is decomposed in the liver into iron, and then the bile pigment, what we call biliverdin. Biliverdin is then converted to bilirubin and excreted. These go through several events until they are broken down eventually into amino acids, and so that's what this is explaining. So this is a picture here of hemoglobin. All right, this is the quaternary structure of hemoglobin. So it is 4 tertiary structures together there. And it's showing you here the iron-containing heme groups. Iron is included within the hemoglobin molecules. And so you can see that structure there. And this is also showing you the difference between the structure of biliverdin and bilirubin, which is important for how we expel this out of the body. Mostly you just need to see the overall structure of hemoglobin. Let's look at the overall life cycle of a red blood cell. They're actually pretty short-lived once they mature. So they start, the small intestine can absorb an essential nutrient, and then the blood transport those nutrients to the ends of long bones, to the red bone marrow over here. All right, so the ends of long bones. In the red bone marrow, red blood cells arise from the less specialized progenitor cells, so they're going to differentiate. They're going to become mature cells once they are released into the bloodstream. All right, and then they're going to circulate for about 120 days. That is as long as they're going to be around. Macrophages will then destroy them, the older red blood cells in the liver and the spleen. Hemoglobin can then be liberated from the red blood cell and is broken down into heme and globin, and then iron from heme returns to red bone marrow to be recycled and reused. And then biliverdin and bilirubin are going to be excreted in bile. Well, these are a very small portion of blood. These are a very important part of our team here. These guys are the ones that help fight off disease and infection. These are our white blood cells, or what we call leukocytes. Leuko means white, and cyte referring to cells. White blood cells, or leukocytes, help protect us from disease. These guys develop from hemocytoblast in the red bone marrow after being stimulated by what we call an interleukin or a colony stimulating factor. Okay, so they basically wake them up and tell them to differentiate. Blood transports the white blood cells to sites of infection where needed. We have 5 types of white blood cells, all right? Neutrophil, eosinophil, basophil, monocyte, and lymphocyte. You need to know these 5 types, and we split these into 2 categories. These are grouped by the nature of their cytoplasm. This is why we call them granulocytes, because some are more granular. And then we have our agranulocytes that do not have obvious granules. And they also are classified by how they stain. All right. And so granulocytes are generally about 2 times the size of a red blood cell. All leukocytes are pretty much quite a bit bigger than red blood cells. And so granulocytes include your neutrophils, your eosinophils, and your basophils. These guys only live about 12 hours, all right? Then our agranulocytes are the lymphocytes and the monocytes. Now these pictures here are not showing any granules, but when you really look at these, they do have a tiny bit of a granular look to them. They're just not as granular looking as the ones that we classify as granulocytes. All right, let's start with the granulocytes. These are neutrophils. These have fine granules that tend to look a little light purple after staining, and you can see in these pictures, none of these are exactly alike, and that is one thing mistake students often make, is they want to look at one and memorize it. Well, you really need to look at the characteristics, okay? So you want to look for light purplish granules after staining. The nucleus is what we call lobed and can have 2 to 5 segments. So here at the top, let me find a good color here. This one you could say has 3 or 4 lobes. This one, you know, is kind of questionable there. Is that 2 things or 1? You can almost even say that's a lobe and make that 5. But you can see that it looks different than this one. This one has about 4 lobes, or segments I should say. So these guys have enzymes that phagocytize bacteria, fungi, and viruses. Do you remember what phagocytize means? Phagocytize means cell eating. So these are guys that go around and clean things up. They make up about 54 to 62% of the leukocytes in the blood. So normally the most common type of leukocyte in the blood is the neutrophil. So if you were doing a blood count and you were counting white blood cells, you would expect to see in a normal sample that these would have the highest percentage. We also sometimes call these polymorphonuclear leukocytes. All right, polymorphonuclear. Poly means many, morph means to change, and nuclear refers to the nucleus. And so these do have different looking nucleuses. This is where we get that different numbers of lobe nuclei here. Okay, they can also sometimes be called bands if they're young, and sometimes they call them segs. So these are often the first to arrive at an infection site. These are going to be a main component of pus, because pus is composed of bacteria, white blood cells, and damaged cells. So while neutrophils are going to be found at a high rate during a bacterial infection, these guys here that we're about to look at are going to be elevated if you have a parasite infestation or an allergic reaction. And it's really important to remember these things, to know what to look for. We want to be able to look at that white blood cell or red blood cell count and say, eosinophils are high, so we've either got a parasite we want to look at or probably an allergy. And so that's the first things diagnostically that are going to need to be looked at. So eosinophils here have very coarse, uniform granules, and these appear more red when stained. They have a bilobed nucleus, so it will be lobed, but there will only be 2 lobes, so that's an important characteristic here. And so these help to moderate or defend against allergic reactions and parasitic worm infections. These will make up about 1 to 3% of the total leukocytes. So the last of our granulocytes are going to be basophils. Basophils are similar in the size and shape of the nuclei to eosinophils, but the key difference here is they have very irregularly shaped granules. So the granules can appear deep blue if you're have a basic stain, and they can often cover the view of the nucleus. And so you can see here, looking at this picture, that these granules do not have a typical appearance to them compared to the previous picture. These are actually going to migrate to a damaged tissue and release histamine or heparin. Histamine promotes inflammation, and heparin helps prevent blood clotting. It inhibits it, so that we have increased blood flow to the injured tissues, bringing all of those components that we need for healing. These are going to be very small in the percentage of our total leukocytes, less than 1%. So this should be the smallest that you're counting as long as things are normal. All right. Hepa means liver, so heparin, that may help you remember that heparin is an anticoagulant secreted by liver cells. All right, so let's look at agranulocytes. Agranulocytes include the monocytes and lymphocytes. Monocytes come from red bone marrow that differentiate into the largest type of blood cell. These are going to be 2 to 3 times larger than a red blood cell. The nuclei is often spherical, kidney-shaped, oval, or lobed, or even this classic horseshoe-shaped. This is what I think of when I think of a monocyte. I think of that kind of a horseshoe-shaped, but it is not always horseshoe-shaped, as you can see in this picture up here. All right. So these are going to leave the bloodstream and then become a macrophage. So monocyte can then become the macrophage once it leaves the bloodstream. That's the distinction. And so when they do, then they will phagocytize bacteria, dead cell, and different debris. These are going to compose about 3 to 9% of total leukocytes, and these guys live for several weeks and even sometimes months. Lymphocytes are our second agranulocyte and the last of our white blood cells. These are a little bit bigger than a red blood cell. They have a large spherical nucleus, and you can see this nucleus takes up the majority of the cell. So these should be some of the easiest for you to identify. All right. Major types of these are going to be T cells and B cells, and those are even broken down further, like we have T helper cells and that type of thing. So these are really important to the immune system. T cells attack microorganisms, things like tumor cells, and even, unfortunately, transplanted cells. B cells are what produce our antibodies, so these guys are vital. Those are proteins that attack foreign molecules and what help us to become immune in the future when we make memory cells later on. So 25 to 33% of all leukocytes are going to be lymphocytes, and these guys can live up to 4 years. Lymphocytes form in the organs of the lymphatic system and the red bone marrow. And of course, their function is to act against a foreign substance. I've already said that about antibodies. All right, so leukocytes need to be able to get to where the damage is. So they are able to squeeze between cells that form the smallest blood vessel. This movement between the cells is called diapedesis, and you can see this here, that they are just squeezing through that tiny little section between cells. All right, and so these lines are showing you these are different cells here put together. Okay, and so then this would be a cell here, and so it's squeezing between those cells. So once it leaves the bloodstream, this guy here will become a macrophage. These are the most mobile and active phagocytic leukocytes. Those are going to be the monocytes and the neutrophils. So this important term here that you need to remember is diapedesis, ped meaning to walk. So what happens when we have a wound? When bacteria invade tissues, like a splinter here puncturing the epidermis, basophils respond by releasing a chemical that actually dilates the blood vessels and makes them what we call leaky. So let's follow this diagram here. And number one, we've got a splinter puncturing the epidermis. Next, after that, the bacteria then can enter because we have broken our first line of defense, that physical barrier of the skin. Once the bacteria get in there, they are able to multiply, and they keep multiplying until something happens to stop them. So injured cells then are going to send out an alarm message. Basically, they're going to release histamine. That histamine is going to dilate the blood vessels, make them larger, and allow more things to flow more fluid, more white blood cells, more stuff that we need basically to repair the damage. And so we say they are leaky. And then the neutrophils are going to move through those blood vessel walls. What do we call that? Diapedesis. And they're going to migrate and actually go to where the bacteria are. And then they're going to be able to phagocytize or cell eat. They're going to be able to eat the bacteria and get rid of them. All right, and so remember, the pus that we see here is made of the bacteria, white blood cells, and the damaged cells. All right, so let's look at some different white blood cell counts. A white blood cell count is a procedure used to count white blood cells or to estimate per cubic millimeter of blood. We typically have 4,500 to 10,000 per millimeter cubed. All right. Leukopenia, these are some abnormal values. So that's the normal value here, all right? So 4,500 to 10,000 millimeters cubed. All right, that's normal. So if you have leukopenia, this is a leuko meaning white. Penia is abnormal low condition. So that would be a low white blood cell count. So that's below 5,000, below the 4,500 mark, really. You will see this with conditions like typhoid fever, flu, measles, chickenpox, and even AIDS. Then we can have leukocytosis. Osis means an abnormal condition. Leuko means white, so leukocytosis means where there is an overproduction of white blood cells. So you would have a very high white blood cell count, so that would be over the 10,000 mark. This is going to be seen with things like acute infections, loss of bodily fluids, and even vigorous exercise, all right? And so to check this, we're going to do what we call a differential white blood cell count, and that is going to list the percentage of the different types of white blood cells so we can look for abnormal values. So this is a good little chart here to review and look at some of the changes in white blood cells that you will see with certain and particular illnesses. So platelets can also be called thrombocytes. Oh, that looks like a little typo there. Thrombocytes. These are cell fragments of megakaryocytes. All right. These are produced by hemocytoplast when thrombopoietin triggers a response. And again, there's that little pathway. These guys don't have a nucleus, and they're about half the size of a red blood cell, maybe even smaller. There's about 150,000 to 450,000 per cubic millimeter of blood, and their function is to help repair broken and damaged blood vessels by actually going and sticking to the surface. When they do that, they release serotonin, which can cause the smooth muscles in the walls of the blood vessels to contract. Now, interestingly, this may actually be the reason why some people that are experiencing emotional distress may cut, because they're actually releasing serotonin, which in the case of a cut, it has a different purpose, but serotonin in the brain is that feel-good chemical. So they're getting an emotional relief actually to cutting. So it may be hard to understand why someone would harm themselves when in distress, but there is a biochemical reason that they're actually getting some relief here. This is a really good summary slide. I would be sure you know everything on this slide. I would even make some flashcards with this materials. Draw them out, especially I'm going to tell you, draw out your white blood cells, because these guys can be hard to differentiate. Many of you are going to go on and take microbiology, and you're going to look at these in much greater depth. And the more you can get those down now, the easier it's going to be when you take micro.